Okay, so my name is William Wen. I'm from University of Michigan. So this particular presentation is supposed to be a project we just wrapped up. The topic is rather specific to most of the statistical genetics audience. However, I think my overall goal is trying to demonstrate the process we solve this type of the particular problem, especially in the presence of big data, large uncertainty, and then in the pursuit of efficient computation. So it's more, maybe the talk maybe sounds more philosophical than practical, but anyway, that's the goal. So I'll give an overview of the problem and then the issues we're trying to address. And then importantly, there's the two parts of the problem solving for this type of complex problem. I would advocate for that. The first part is called model building. It's more or less related to create a generative model, probabilistic generative model. And then the second part of necessary in that sequence is to think of how to use the existing tool to trying to fit the complicated model. So this is more like computational or practical consideration for solving this particular problem. So I will demonstrate a numerical example and give you some summary remarks. So overview. So everybody works in these computational genomics. We're in the big data. Actually, to say that there's a big data is an understatement. We have, for example, for genetics, we have millions of SNPs, thousands of phenotypes. That's the expression levels you measure from tens of thousands of genes per individual sample. So that's a huge data problem. And nowadays, we encounter multiple data sources. We have genotype data, we have genomic annotations, we have molecular phenotypes, and then at the end of the chain, we have the organism level phenotypes like complex trees and disease status. So with all these things, complex things presented, I think there are two considerable, actually difficult issues you may or may not appreciate. One is you need to formulate a sensible statistical problems. So the common practice in the field is trying to formulate everything into hypothesis testing. The rationale is natural. Everything is, we are doing hypothesis-driven science. So why not statistically, we just solve the hypothesis testing problem. In reality, you will find nowadays, especially in the presence of like multiple data sources, complicated data sources, hypothesis testing, if you start from that angle, may not make the solution easy. So hopefully I would convince you through this presentation. The second part is efficient computing. So you can think of a very complicated, elegant model, but if you don't have a method to fit it, to get anything out of it is useless. So the overall message is you have to achieve a balance between the formulation of this statistical question by constructing a reasonable model and to think of efficient computing to feed the model. So the particular problem I'm going to discuss is related to the so-called co-localization problem. But just a little bit of background, what kind of the data are involved in this problem. So there is a simple kind of a biological model. So we have genetic variants. It's probably immediately going to impact on the molecular phenotypes, that is gene expression. And then those effects are accumulated and then eventually lead to some organismal level changes. So this is simple kind of biological model everybody seems like accepted. And so this may or may not be true. So that's why we do data analysis, trying to pinpoint for the detailed examples to figure out if that's actually the case for the observed data. The important thing to note is that if we collect the data from this part, and this part, the data itself naturally form a hierarchy, right? I mean, just like I said, the hypothesis is saying there is a hierarchy. And so that's hopefully kind of already persuade you towards the thinking of hierarchical model, okay? So the general motivation to put everything together to do the data analysis, there are twofolds, obviously at least two. There's 2 points. One is by explicitly integrate all the data, you may be able to explore the underlying biological mechanism. For example, if this model is right, or there's some alternative explanations of the you observed the data. The secondly is when bring in some extra information, for example, gene expression, even your goal is trying to find GWAS hits. This extra information is supposed to help you to gain power. So that's the simple motivation. So this slide shows the specific problem we have in mind. So there's more detailed statement about that problem. So suppose you have already done the GWAS, or you have already mapped the EQTLs. By the way, the EQTL is just simply the genetic variant associated with gene expression traits. So you want to, at least at the very basic level, you're trying to assess the overlapping of the two associations, right? But the goal here is identify the genetic variants which are both causal. So the keywords here is a causal, EQTL, and then causal GWAS hit. Well, I mean, that's the goal. You may not get there because, as I said, there's observational data. You may not be able to pinpoint if that's really the causal EQTL, and then that's the causal GWAS hit. But with some sort of the statement, you should be able to give an assessment if there's in this particular locus, there's two GWAS, the GWAS hits and the causal EQTL are actually overlapping. So that's the statement of the problem. And in the process of solving this problem, I argue you need to solve two related problems. The first one is we call enrichment problem. You have to have some sort of quantification of enrichment level of causal EQTLs in the causal GWAS hits. I'll demonstrate this through a few examples, maybe not obvious at this point. The second point here is you need to do a very good job in terms of fine mapping GWAS loci, accounting for the EQTL status. Is that clear? So if you have questions, just, so this is very important to understand the problem we're trying to solve here. Okay, so you may already have something like algorithm to do this and this in your mind. I'm just trying to do the approach I was advocating a few minutes ago. So first of all, I'm going to write down a probabilistic generative model to describe how my observed data should look like. So this is the primary scientific goal, right? This is probably redundant. Sorry. So that's related to the model building and then formulation step. So this is the process you build your probabilistic generative model. The key here is you ask yourself how the data generated with noise, obviously. So you want to write down a model that you can use to do realistic simulation. So in this particular problem, we can consider a complex trace. So that's the GWAS part. And then we just model that with a vector of, it's a vector y here. And then I'm going to use a linear regression model to correlate with all the genotypes, all the P genotypes. So that could be millions. At this point, I don't really concerned about the computation, okay? Just thinking about how to describe this kind of association. So obviously, this is going to be a very high-dimensional regression model. So each beta here gonna characterize the effect size of the association. As we know, most of the SNPs gonna have beta equal to 0, and then some of the really interesting things has beta is non-zero. So that's why I argue the real point of interest here is this latent binary variable I defined as gamma. So if gamma equal to 0, that's not association, not a causal association actually. This is a little bit more than just observed association. If beta is non-zero, then gamma equal to 1, that's the true association. And then I would argue that the true interest will be identify this latent, very high dimensional vector gamma with OP SNPs. Nowadays, P could get to 6 million. So this is if you stop right there, this is already kind of a known statistical problem called as a variable selection problem, right? You just need to figure out what gamma is. The important argument here is the beta is not really important here. It's mostly treated as a nuisance parameter. I would argue if you're really interested in beta, the first consideration is the question you need to ask yourself is why do you use a linear model? Why do you believe additive effect and so on and so forth? So we don't take that very seriously. And we're really trying to define the robust kind of a discovery of association. So even this model is not quite right, it still can give you the robust identification of causal association. So that's why gamma is more important than beta. It's a simple argument. To incorporate the known genomic annotations, so you have SNP level annotations nowadays. You can have transcription factor binding site information. You may have methylation information, so on and so forth, a lot of molecular level annotations. You can easily incorporate through a so-called prior. It's actually one level up. It's like a hierarchical model. So a priori, you may just model the probability of a particular SNP being associated. So that's probability is associated with the feature you have. That's the annotation. So that's alpha 0 and alpha 1. This is a very simple logistic model. So if the alpha 1 is positive, meaning that if you have a feature, let's say the feature is 0 or 1, it's the binary annotation. If you have the feature that will increase the odds of a particular SNP being associated, right? Simple as that. The second line is just a priori. You think everything is independent. It's very important to stress this a priori assumption. And then this alpha parameter would be called enrichment parameters throughout the talk. And then that's very important, right? So through this model, now you already have the ability to kind of simulate your observed data, right? If you have gammas, you can use this particular model to simulate gammas. This is just Bernoulli's with different parameters, right? Determined by the features. And once you got the gammas, you just know how to simulate beta, then you get this type of the thing. This linear model will help you to simulate either the complete data or the summary level statistic. So that's why this is called a hierarchical model, but it's also a probabilistic generative model. Our problem is a little bit different. So this is this is more like a solve the problem used in a lot of approaches, I would say. But the problem we have in mind is a little bit different. So this whole framework is natural to incorporate EQTL status as a notation, right? I mean, if I tell you that is a known thing, the EQTL status, so either it's associated or not associated, so there's kind of a binary annotation, then we're done. We can just use the previous model. Nevertheless, we have to notice that the EQTL is also kind of mapped as association studies, right? As I said, EQTL is identified through association. There are typically, there's a lot of uncertainties associated with that, the annotation. due to either insufficient power or the linkage disequilibrium and other factors. So you are not really confident to say at the particular SNP is a 0 or 1, it's a EQTL or not a EQTL. You maybe say this SNP is likely to be EQTL, but you cannot also exclude its, for example, its LD partners. So in light of this, the better way to describe our data is actually think the delta is also latent, right? If delta is observed, then it's just plug in here. But the delta, if delta is latent, we kind of think the delta is kind of a draw from this particular posterior probability. It's in the form of the posterior probability. So it's, if you analyze your data in the Bayesian way, then you're going to get this whole posterior distribution, and then you're going to get this delta account for the uncertainty caused by all these factors. So that's really the key of that in terms of modeling we added to this particular problem. Now, with this whole model set up, now we get to the stage of formulation of the problem. Then it becomes very straightforward or very natural. If you want to assess if a particular SNP is that particular SNP, if an EQTL and then the GWAS hit overlap, you just simply ask this question, right? This is the probability statement about overlapping. So at this particular SNP, gamma I equal to 1, and then delta I equal to 1, so that both causal EQTL and then causal GWAS, given all the observed data. So supposedly you should be able to solve this because you have the complete model. And to compute the whole computation, you will also notice would require the inference of the alpha parameter, the enrichment parameter, which is essentially this probability, given the delta, given the SNP is EQTL or not EQTL, what is the probability that particular SNP is a GWAS hit. So get to this point, we're a very clear goal, right? I mean, we set up the model, and then we just need to fit the model, figure out these things, that's it. So the next few slides, just trying to convince you there's the importance of this second quantity to the overall approach. So we need to, so this enrichment parameter essentially is trying to quantify the dependence or independence between the EQTL annotation and then the genetic association of complex traits. So there are some extreme examples that maybe make a lot of sense or to you. If you assume this quantity, the delta I, and then the gamma I, now you really can treat them as random variables. They're independent. Then you have this kind of the equation you have in mind, right? So there's a given delta, and then the status has nothing to do with a particular SNP being a causal EQTL. The same thing here. This is another extreme. If you assume delta equal to 1 will lead to gamma I equal to 1, this is essentially assume all the EQTLs are going to be causal GWAS hit. So that's basically another level of extremeness. So it's basically saying you would expect all the EQTL you have has something to do with the upper level phenotype. So that's what we call the maximum enrichment. So even without any data, I'm arguing that you use this type of the model, you can already do something. And in particular is calculate your expectation. So what this is a simple interpretation or translation from that logistic model. So this row here is defined the proportion of GWAS hit going to be overlapped with your EQTL data. So if you say 50% of your GWAS hit going to be overlapped with, going to be causal EQTL, then rho equal to 0.5, obviously. So this, the right-hand side related two things. One is this alpha parameter. This is the enrichment parameter. The other thing is basically the prevalence of EQTL in your data. So it's related to the ability to identify EQTLs, what kind of EQTLs. So that's the P of D. That's basically proportion of the SNPs are causal EQTLs. So the GTEX data basically is a very good resource to get the P of D, I would say, argue that. So this is actually something from real life example. So we have this grant, and at the stage of funding, so the NIH say, you need to cut your sample, especially doing the EQTLs. And we said no, we need that sample size. So this is basically the sample size to do the EQTLs. We proposed to do 150, but they say you can cut to under 50 because the argument is, see, there's such a study, they do 50 EQTL samples, they still find plenty of EQTLs. Well, that's basically true, but at the same time, if your goal is overall integrative analysis, it's not just the pure amount of EQTLs you find, it's more or less how comprehensive the whole set of the EQTLs you can find. So this is basically demonstrate that, so we need to build this curve. So each individual curve basically is what we learned from the GTEX data, what proportion corresponding to different P of D, the prevalence of EQTLs you can find. Obviously, with larger sample size, you find more. The x-axis here is actually alpha 1, so that's different enrichment level, and the y-axis is 0 here. The point here is, if the enrichment level is very low, like in the 0, that's the independent case, or 2, you shouldn't expect to find anything, well, anything extraordinary. Like you may find 1 or 2, that's very normal. If the enrichment level is very high, even you have small sample size, you should find plenty of overlapping. I can tell you now, actually, estimation from the data that's basically based on the later approaches is actually in this area. It's 405. It's actually a very large odds ratio. Think about this. So the sample size does make a very big difference if you have this. So the whole demonstration here is trying to say the sample size matters, but I mean, the side note is even you have a prior model, you don't have any real data, you can do some sort of the projection and then trying to understand, give yourself anticipation what level of co-localization you're going to find. Now you have the whole model. The goal is trying to feed the model and then get something useful. So usually, if you have a Bayesian model, people are going to say, all right, you have the Bayesian model, you should run MCMC on it. But this is a particular issue here is we have a scalability issue. So MCMC is expensive and not scalable. But think about this. We have this is related to the sampling of the gamma parameter. Sorry, I shouldn't go back. The gamma has like 6 million SNPs. So the sample space is astronomical, right? 2 to the 6 million. There is no way any sampler can survey the field very, very well. So you have to find alternatives. So we kind of think about this problem and go back to the literature, and then we kind of come up with this type of the solution. So there's different levels of the approximation we're going to do. So basically, you cannot do the generic thing. You cannot get the exact solution. So you need to think about more or less the approximation approach. So the thing we come up with, basically summarized in this equation, there's actually packed a lot of information. The first thing is we're going to chop the genome into small chunks. They're called LD blocks. We would basically show this posterior probability of this quantity we're looking for is approximately the product of that. This is due to the relative independence of the SNP in the block, between the blocks. Secondly, is also a consequence of we have a priori independent prior. The second thing is more important. So this side, you need to do an integration to get rid of alpha. So this side is an integration. This side is it's actually maximization. So we are using so-called parametric empirical base. Substituted the best value, some sort of the best value of alpha into it. So that's translate certain level of integration problem to a maximization problem. So the argument of that is actually very interesting. We go back to the literature, and then we find there is something interesting about that. So this is so-called Laplace transformation for Bayesian inference in conditional independent hierarchical model. So this is a kind of a 1988, 89, when people don't have those powerful computers, they think about these computational tricks. And then the argument is they're actually very useful nowadays. So look at this. So the statement is give you not only saying the right-hand side is is approximation of the right-hand side, also give you an arrow bound. The important thing to notice that the P here will be the number of the LD blocks in our context. And then this thing is, this arrow term is a relative arrow. And then it's so much better when you have a relative arrow to basically estimate a small probability, right? In contrast, if you do MCMC, any Monte Carlo based method, you're gonna get additive arrow. So if your quantity itself is small, and then you have additive arrow added to it, then you need tremendous amount of MCMC run, basically, to reduce this term. But this, from kind of a mathematical thinking, saying whatever you do, you get a relative arrow. So in terms of absolute terms, if the quantity you're trying to estimate is small, then automatically the arrow is going to be small as well if you stick to this. So very useful thing. Just saying there's the old literatures are still helpful in nowadays. So with this, now we're just trying to solving a classic missing data type of the problem, right? Because the gamma L is latent, it's not observed. So in the statistical literature, we know that when you have a missing data problem, the first thing you do is trying to see if the EM algorithm solve it. In this case, yes, in principle. You have an E step trying to calculate this. I'm not saying this is easy to calculate, but that's what they need. And then in the M step, it's really easy. You just got the fitted PIP, so this is a posterior inclusion probability calculated in the previous step, and then regress on the regression. And then that regression coefficient is actually maximize the alpha. So that's pretty straightforward. The only thing left is trying to compute the gamma, how to calculate this quantity, which I will say that a little bit later. There are additional challenges in enrichment estimation. So the EQTL annotation is intrinsically imprecise, as we discussed, and only know up to a distribution. That's based on our model. So instead of just classify 0, 1 according to the probability you have behind, the better solution is actually already known to the statisticians in the field, like survey sampling and maybe biostatistical, like survival analysis in this field. This is called multiple imputation. The idea is very simple. This is kind of a stochastic approximation. So if you have this kind of a distribution, just sample from this, create pseudo data sets with all the missing entries filled. And then you just analyze this data, see if they are actually observed. At the end of the day, you just combine from the multiple imputed data set. So this is a kind of a well-established theory called the multiple imputations. Notice that the use of the imputation is a little bit different than we use in the imputation in the statistical genetics. So when this is done, you can estimate alpha hat, alpha 1. Then the only thing you need to do is construct a so-called mixture prior for accounting for the EQTL data, right? So you have the probability that a particular SNP being an EQTL, which is this probability, and then with the probability, this probability, and then they have different odds of being causal to the GWAS hit. And next thing I'm gonna say, this is the thing I'm going to hand-wavingly say. Then you use a different algorithm trying to estimate this PIP, the gamma I EQTL data given GWAS data. So we use this algorithm, actually our self-made algorithm, so-called deterministic approximation of posteriors. It's actually a kind of a good solution for this type of the variable selection in relatively high dimension. So if you have 3,000 to 4,000 SNPs, you can easily apply that and then give you accurate result. I'll show you an example a little bit later on. And once you get this value, at the SNP level, you can easily calculate this, the desired quantity you set up to pursue very easily by solving a simple linear system. So this is trying to demonstrate why we do basically DAB instead of MCMC. So this is a simulated data set. Each data set has only 15 SNPs, but we repeat that with basically 1,000 LD block. The reason we select the 15 blocks is because when you have 15 SNPs, you have about 32,000 configurations. You can compute them, basically enumerate all configurations. So you get sort of the exact solution. So on the one side, you want to run MCMC. So you have 32,000. So we run MCMC. This is a kind of a vanilla version of the MCMC. It's more like select configurations in a random walk fashion. So we run 1 million MCMC for 15 SNPs. So this is the results. It's not surprising. So you will see the MCMC doesn't put the weights equally on every level of association. So this is undersampled. On average, every configuration should be visit like 3 times, but now not equally. So there is a lot of things in the middle undersampled. Under the same setting, if you run the DAP, it's not perfect, but you get more or less, actually you get more accurate in the sense that the overall accuracy is higher. Most importantly, this is just using a fraction of the MCMC computational time. So that's really kind of a desirable in this particular setting. There is a more practical issue in the summarizing the results. What we have there is a SNP level co-localization. But I would argue in this particular context, even at the SNP level, you may not be able to fully distinguish the LD. Therefore, report SNP level co-localization probability may not be the best thing you could do. On the other hand, if you have a region with LD, our solution is actually sum over all the SNP level inclusion probability, because presumably they are kind of represent the same unobserved the causal association, and then call that as a regional co-localization probability. It's more meaningful. So give you an example. So if you have 2 SNPs in perfect LD, and then I tell you with probability 1, there is 1 SNP is causally associated with the GWAS. And then with probability 1, there is 1 SNP that causally associated with the EQTL. Well, it's a causal EQTL. Then I ask the question, what is the probability that this region, this 2 SNP, contains co-localized signal? Give you 5 seconds to think. All right, the answer is depends on the enrichment parameter, right? So you can draw this very simple. If you think they are just basically independent, there's 2 things not related at all, then there's a 50-50% chance they are overlap, right? And then if you have a high enrichment, then more or less you're going to say there's a close to the 1 end. So again, this is kind of the trajectory here is saying they're sensitive to the actual enrichment parameter. That's why you could, the enrichment estimation is particularly important here. The other thing to say is, once you have that, if you can say something about the whole region, right? If you have, for example, you have an enrichment parameter here, you say, okay, the co-localization at the region is close to 1. But keep in mind, you still don't know which SNP is causal. meaning the two signals are overlapping. But that's the nature of the data. They are observational data. They're in the statistical terms, the two SNPs are not identifiable. Okay, so the last bit of the advantage of this thing, maybe in the end, you come back to this question. I want to do some statistical testing on the co-localization. Can I do it? Yes, if you're Bayesian, you could do Bayesian FDR control. That's actually very simple. If you have that probability, 1 minus that probability is actually false discovery probability. And then the average of the false discovery probability is actually the false discovery rate. So this is mostly built on the work by Michael Newton and then Brad Efron. So if you have probability, it's very easy to translate into a hypothesis testing problem and actually solve for it. On the other hand, if you're trying to solve it in a frequentist way, to set up a null hypothesis saying, okay, maybe there's not overlap, then you have a compound situation, right? Because this is really complex null hypothesis. You may have one SNP is EQTL, one SNP, but they are not overlapping, or you may have just one signal, or you may have none signal at all. So there's a compound situation. So I don't know any good statistical frequentist solution, but if you have one, let me know. So I'm going to show you some numerical examples. I'm going to be a little bit faster in this stage. So you cannot read this plot. This is based on simulation. The point here is you can now see there's actually 5 different curves. They all kind of get together. So this is the ROC curve. There's different settings. One is using individual level data, and then the first one is using individual level genotype data, and then using the true enrichment parameter. The second one is individual level data, but using estimated. So we do the multiple imputation. The third one is using summary level GWAS data, and then the true prior. And then the next one is summary level data estimated prior. The last one is you can actually assume that the two things, the enrichment parameters are independent by using individual level data. So in the ROC curve space, everything got very close. And then you think about this, that actually makes sense. This is about how you're ranking your co-localization signal. So that basically saying, even you do this very complicated statistical modeling, at the end of the day, the ranking is rather consistent, right? If you change the ranking, that will change the ROC curve. But if you transform in the monotonic way, your PIP or whatever the quantity you are doing, that doesn't change the ROC curve. But in practice, though, you need to find out what is the true FDP, right? This is the false discovery rate. The argument there is if you do this type of the sophisticated model, the benefit is a better quantification of uncertainty. So this is showing if you actually do the hypothesis testing, then this 4 method are going to show a little bit difference. So knowingly, the last one is you assume everything going to be independent. So it's very conservative. First of all, the truth is about, we use the truth, the alpha is about 5. Same thing here. If you look at the power, this is really kind of a quantify the probability. Then you see this is a poor quantification, but doesn't mean it doesn't rank the SNP very poorly. So you have to determine what is your want, right? And you want the full quantification in a very statistical rigorous way, or you want just generate a priority list. If you do want to just generate a priority list, then maybe this tells you could just use that independent example. So finally, a real data example, but I'm not going to say too much about it. So this is the GWAS data come from the global lipids, and the EQTL data is come from the GTEX whole blood. But there's a intermediate release with a limited sample size. So our main findings you can read from this PLOS genetics paper. And then the software with source code, and then the real data example. So everything data is public, so we just included the data example in the GitHub. So you can replicate our findings, see if we made any mistake. So overall, we identify kind of 18 genomic regions, and then there's nothing out of ordinary. So the point of doing this is kind of a reassuring. Remember, the original global lipid study, they go very, very far. This is a very, very good work, but doing by their the biologists and then the statistical geneticists. They go out to collect all kinds of the source, trying to pinpoint what is the relevant gene, and then in our kind of analysis, we kind of confirm their findings are actually right, but with only the EQTL data, though. Okay, I'll just summarize. Then I will just try to answer the question. The most important message here is the conceptual separation of generative model and then fitting algorithm. It's very, very helpful in terms of tackling big data problem. If you mash the two things together, I think it's not dual. I think in a certain level, you will either suffer a practical problem, meaning that you cannot do the computation, or you suffer a problem like statistical rigor problem. Okay, so you have to balance the two. So the Bayesian model is conceptually very easy. However, inference are computationally challenging. So you may go out of the traditional way to think maybe MCMC is not the only solution. You need to take some context-dependent information and then trying to find a solution that's feasible to your problem. So instead of using a generic tool, you go with the you do a little bit context-dependent innovation. Right, so that's probably, this is a summarize of this so-called no free lunch principle. You could have you need to find the efficient, accurate approximation is the key. You cannot do very sophisticated model, just work on that without considering computation. And then at the same time, you shouldn't just find a solution, just simply you can compute without, ignoring the statistical rigor. So finding the balance is really the key. I think that's all I have. I have to thank, I thank my students and collaborators, and then this GTEX consortium, and then the Global Lipids Consortium. Thank you very much.